So let me get us started. This tutorial on SBOL data model version 3. So I want to start with just some sort of general introduction to the notion of SBOL and what are the pieces of SBOL. For some of you, this may be familiar. For some of you, it may be all new. SBOL, Synthetic Biology Open Language, is actually two coupled standards, each of which can be used alone, but which reinforce one another nicely. One is the SBOL data model, which is a formal representation of objects that we'd like to describe having to do with design, build, test, learn in synthetic biology workflows. And it turns out that because synthetic biology is so cross-disciplinary, you can use this for a lot of non-synthetic biology workflows too. A lot of what you do in any sort of molecular or cellular biology or even chemistry turns out to be representable in terms of SBOL workflows. That's the focus of the tutorial today. There is also an SBOL visual standard, which you heard Goxel talking about, one of the associated artifacts this morning. And there is a tutorial for that on Friday focused on how to visualize genetic designs using graphical symbols. Both of these do not exist in a vacuum. As much as possible in the design of SBOL, the goal has been not to reinvent the wheel because there's a lot of great bioinformatic resources out there. So to a very high degree, SBOL leverages existing ontologies like the sequence ontology, the systems biology ontology, the measurement ontology, the provenance ontology, etc., to handle portions of the representation. Now, this venture of building a language like this has been going on for a good while, at least, kicking off more than a decade ago. I'm not going to go through all these milestones in detail, but just to note that what we're talking about today is something that has been evolving in the hands of many different people at many different organizations for a good while. I am going to talk about the history in a more comparative way by talking about how SBOL compares to other types of representations of biological design that you may be more familiar with. So, your basic world where most genetic design is encoded and transferred back and forth is FASTA and GenBank. FASTA, you've just got your sequence. GenBank, you get a little bit of labeling on the sequence. These are great for if you are studying existing natural systems and you want to say, okay, what did I find here? But they're really not adequate if you're trying to do engineering, where you have a lot more to say about the structure. SBOL 1 gave us the first piece of that. It's also, if you're familiar with GFF3, GFF3 is sort of halfway between GenBank and SBOL 1. SBOL 1 lets us talk not just about a sequence with some features, but a hierarchical structure, and a structure in which we might be only partly through the design process. We may not have made all the decisions about what's in our design and where are some things that we still need to make choices about. That was all well and good, but one of the other big pain points is that in a lot of cases, the thing we're studying isn't really the genes. We're studying what the genes do. We care about their regulatory interactions. We care about the things they make. We care about the things that trigger them. So SBOL 2, really the big leap there was to move from just talking about structure to talking about both structure and function. So be able to represent the products, their regulatory interactions, their reactions with one another, etc. And that meant also being able to represent proteins, small molecules, other sorts of compounds. And at the same time, SBOL 2 also brought in the ability to start talking about design libraries. So like the keynote that we just heard had a lot of talk about making complex libraries with many elements in them. In order to be able to describe and do that, you need some way to describe the whole library where you haven't necessarily built all of the instances of it yet. And SBOL 2 introduced a notion of a combinatorial derivation, which describes how you build a many-way library like that. That got us a good long way, but there are a lot of little awkwardnesses. And SBOL 3 essentially was the result of taking all the things that had happened so far and a whole bunch of pragmatic experiences applying these models in projects and boiling it down to have simpler and more succinct models, while also extending the hooks to link throughout the design, build, test, learn cycle. So this is the general idea of what we're going to talk about today. I'm going to go through some specific examples, building up each of the elements in turn as we go through. So in particular, we're going to focus on the data model. I'm going to explain all the pieces of this in more detail later. Right now, I just want to show you that this is basically the set of things that we talk about. Functions, the blue classes, with everything sort of hooking to this core hub of the notion of component. And then there's another set of classes that extend up and down the design, build, test, learn cycle, which are generally more hooks for connecting to other things and less complete descriptions in and of themselves. SBOL visual, complementary to this, lets us show everything in the diagram. So SBOL visual lets you make the diagram. SBOL data lets you represent everything that's in that figure in a paper. You'll hear about this on Friday if you choose to go there. SBOL Visual 3 has just been released. It's essentially identical to SBOL Visual 2, but it talks about how to relate to an SBOL 3 data model as well. So the reason why we want this is because we want to be able to hook together different types of tools all the way through the design, build, test, learn pipeline. We need to be able to talk about design tools, talk about how to get from there through various choices about mapping onto specific parts, onto the manufacturing, quality control, getting measurements in, and how we connect even to the models out of that. And so in between all of these pieces, we end up having data repositories of various sorts. I'm just going to breeze through a few examples of tools out there that use SBOL. Most of these are still SBOL 2 tools, but some of them support SBOL 3 as well. So an example, the SBOL ME data repository is a metabolic engineering focused repository of information encoded in the SBOL 2 format. ICE out of JBay LBNL is a repository that talks SBOL as one of its languages. SynBioHub is a data store backed with a triple store that lets you easily publish and browse material in SBOL format. You heard a little bit of that this morning when Yet and AJ were talking about the Excel to SBOL plugin for uploading into SynBioHub. And SynBioHub still primarily SBOL 2, but it can also ingest SBOL 3 materials. And another key approach in repositories that SBOL 3 has enabled is to just forget all the specialized repositories and just store stuff in Git, because SBOL 3 has visualizations that are, serializations that are version control compatible. Benchling is an example of a sequence editor that talk some SBOL, although their support isn't great. It was really good at one point, but it's decayed over time as they've gone more walled garden. They don't really speak GenBank correctly either at this point, unfortunately. SBOL Designer is another editor, and a more recent one, SBOL Canvas, is out that lets you show the links as with functional links as well. Folks may be familiar with the Voight Lab design tool Cello. So Chris Voight, Doug Densmore, this system for designing genetic logic circuits that encodes material in SBOL in order to link the designs with the models. SBOL talks with the systems biology modeling language through tools like iBiosim and connects to that whole modeling side of the world. So that's just a sample of a few of the types of tools that are out there. I'll briefly note that how SBOL gets developed. There's a broad community of people who just sign up to be on the announced list and get occasional newsletters about what's going on with SBOL. People who actually want to be involved in the development. It's recommended that they join the SBOL Dev mailing list, or you can just start showing up on GitHub where all the stuff is done out in the open and start commenting on issues on repositories. The leadership of SBOL is an elected chair and an elected set of editors who serve as essentially community managers and curators of the standard. And they get supported by a steering committee and advisory panel. And there's also an SBOL industrial user group with a number of companies in it. I'll just note that as we go along. And you've heard some of the work already presented today was supported partly by the Industrial Consortium. The Industrial Consortium is both investing in SBOL and coordinating with regards to their use of SBOL. So if you're interested in SBOL, the best way to get involved is just to use it. And then the next thing is if and when, more likely when than if, you run into challenges while using it, report issues and get help rather than just walking away from the tools. If you want to get more involved, then you can get onto those mailing lists. If you're at a company, bring your company into industrial. And of course, you can get deeply involved in an open project if you want. So that's all of my prefatory material. I want to spend the rest of this discussion focusing on the data model itself and giving a walkthrough there. But before I go that, let me just pause a moment and see if anybody's got any questions about the context that I just talked through. All right, hearing nothing, I'm going to dive into the data model itself. And I'm going to remind, we're going to start out at this core. Component is really the core that we build everything around. Component is what you use to represent a cell. It's what you use to represent a piece of DNA. It's what you use to represent a protein. It's what you use to represent the design for an experimental sample. So component is our sort of source of all of most nouns. If a description of a thing in a lab is going to be a component. And we're going to have these blue classes talk about the functional nature of that component, what it does. And the red classes going to talk about the structural nature, what it's composed of. Now, component is essentially a design element, and it links out to the actual sequences that we may be describing of of DNA, RNA, protein, serialized versions of chemical structures for small molecules. Combinatorial derivations are how we describe the design of a library. Models are essentially link outs to models in any other platform. And this is all in the sort of digital world. When you actually go into the lab, if you want to talk about a specific aliquot in your lab or a specific replicate in an experiment, that's what implementations are for. And then once we've done something in the lab and gotten some data, then experimental data essentially lets us give pointers to blobs of data that we want to associate back, collect those together in experiment objects, and then we stitch these all together with provenance elements like activities, and have some sort of meta level classes like collections for just making bags of stuff. That's our sort of equivalent of a file folder, and attachments for putting pointers to stuff of any indeterminate file type out there in the world. So we're going to go through each of these in turn. But first, a little technical preliminary. What lies underneath the SBOL data model is RDF. RDF, the Resource Description Framework. I have to look that up every time because really people just tend to refer to it as RDF. RDF is the foundational linked data language, the most commonly used one in the bio world. Over in the AI world, there's a bunch of competing linked data languages, of which RDF is one. But if you're in the biomedical world, RDF is pretty dominant. And it basically, all these graph languages basically let you talk about things in terms of sort of subject, verb, object statements called triples. RDF is one way of doing this. So we can say things like, this particular plasmid has a feature on it, which is this particular eco-RI cut site. Or this particular cell strain contains that plasmid. Or this flow cytometry file that I have here was derived from this sample. The nice thing about this triple model is that in theory, it's universal and can store any type of data or knowledge. It turns out everything can be potentially boiled down to triples like this. It's also because there is so much use of this in the biomedical world in general, it integrates well. And part of the enabler for all this is that the model that RDF uses is an open and extensible model. You can always add more words to your vocabulary without conflicting with the existing words. This is something that differs from stuff like, for example, JSON schema, which is not readily extensible. You pick a schema and you get kind of stuck in that schema sometimes. There's a lot of well-established free and open tooling for this, including a lot of nice libraries for working with it in a lot of different languages. And there's a standardized serialization to a bunch of different formats, including XML and JSON. And for my purposes, the one that I am happiest about is there's a plain triples format, which when you sort it, makes your life really nice in version control because you get a stable format every time, and you can look at diffs really cleanly. What makes all this work is that these subject, verb, object triples, the subject, the verb, and the object are always URIs or IRIs. That is unique names for the objects. The most typical form that you have seen URIs in is URLs. So your web address URLs, that's a class of URI. UUIDs, if you encounter them, that's another class of a URI. So this essentially gives us a way, just the same way that you do with setting up anything online on the internet. You have a unique naming approach, and you can know which portions of that namespace you control and which portions somebody else controls and are not under your control. So it's very easy to ensure that things under your control are uniquely named. And SBOL has some suggestions about how to structure your URIs to help make sure that you don't end up with collisions. We define our data types based on the RDF gives a standard sort of bootstrapping up in terms of strings, numbers, either integer or floating point, Booleans, and URIs. And then you can put those together into classes. And you're going to see a bunch of class diagrams that have this sort of look to them, where you've got a class name, properties with numbers next to them that talk about how many elements you can have of that property. So if it's got a 1, that's a property that's required. You must have precisely 1 of this. If something's optional, then it has a cardinality of 0 or 1. If you can have as many of them as you want, like say how many members you have in a collection, then is the 0 to star. It's an unbounded property. And you can have it be both required and unbounded. Like you have to have at least 1, but you can have more than 1. These little arrows will show subclass relationships. And then when we reference objects, some objects are bundled inside of another object as a child object. That's something that really doesn't make sense to talk about on its own. So if I'm talking about a cut site on a plasmid, that cut site doesn't really exist separate from the plasmid. It doesn't make sense to say EcoRI cuts here if I'm not talking about that as part of the plasmid. Other ones, other elements we reference rather than including them in. That's, for example, if I talk about my plasmid has a copy of a GFP coding sequence on it, the GFP coding sequence exists independent of the fact that I happen to be including it in this particular plasmid. I see a question, what happens if you experience URI collisions? So this actually brings us to why I love the fact that we can do version control. Because if you are working in a Git sort of environment for managing your SBOL, then you can see a URI collision show up because you end up with something being defined as having conflicting information in it. And so it shows up as a conflict, just like any other code conflict. If you're managing it in other ways, you may or may not detect it quite so easily. How to avoid getting collisions then brings us right back to sort of the same way that we manage naming in any software world, where you coin names in the spaces that you control, and you do not coin names in spaces that are not part of your project. If you create a collision, then under the rules that we've got in the particular libraries, your library, if you add another object with the same name, that library is going to throw an exception at you. What you do with that then becomes a question for your particular use case and your tools. Do you rename it? Do you have a merge? Do you go and yell at somebody who put something in the code base that they shouldn't have? All of those are potentially viable options for resolving a merge conflict. The important part is that you can detect it early. Did that answer that question? All right. So a little more about URIs. This is very good lead in here. So the two root classes for everything in SBOL are top level and identified. Every object is an identified object, and that basically gives us an identity and then some metadata hook points for it. So we have a short form of that identity. So think about if the identity is the full path name of your file. Then the display ID is the name just of the file. And the name is the human-friendly version. What do you actually want to show somebody if you show it on a diagram or on a report somewhere? Description is just what it sounds like. You can add an arbitrary pile of text describing anything however you like. And then there are some hooks for provenance information talking about where this thing came from, if you wish. And we'll talk about that more later. Everything's unidentified. Top level subclasses are things that stand on their own. Top level is something like a plasmid, a coding sequence, a strain. Child objects are nested inside of top levels. Those are things like like a feature for a cut site, or the interaction between a genetic production interaction between the coding sequence and the protein it produces. It doesn't stand alone. So it's a child object. We'll see those as we go along. SBOL 2 was pretty loose about what you could do with your URIs. Your URLs could be pretty much anything you wanted. SBOL 3 is a bit more specific about, if you're using URLs, how to structure them. And it structures them basically using the sort of file tree-like structure that you see in typical URLs. You've got a namespace, and that's the area where you want to think, I have control of this. Everything in my blob of stuff in my namespace should live in kind of one location where I can look for conflicts if they're going to emerge. Your display ID is the last element. And in between, however many layers of sort of directory structure you've got, that's the local between the namespace and the display ID. So think of it as a namespace gives you a little area of file system that you're naming things within. So for example, here's an iGEM part on SynBioHub, this J23070 part, which is synbiohub.org is our namespace that we're working within, and then there's a local here. In response to the question, people should ask questions as they have questions in the chat. This is your tutorial. It's not about me. It's about getting your questions answered. When you've got a child object, then you add more layers after the parent. A child object always must be prefixed by the name of its parent. Having this structure makes it easier to control and track things and make sure that when we're transferring an object from one place to another, we can transfer all the stuff that goes with that object. So,
Jake, do you mind, can you dive in a tiny bit more into the difference between here, the namespace and the local part of the URI? So here you have SynBioHub, and then you have iGEM. So one way you could think of it is that, igem.org would have been the namespace for the iGEM parts, but here it's synbiohub.org. Could you say a little bit more about how you choose what is going to go where? Sure.
So your URL, if you're using URLs and not UUIDs or some other thing, but most of the work goes with URLs. So I'm just going to assume URLs when we're talking. Your URL should be a thing that if you follow this thing, you should be able to find the SBOL object that you're looking for. Now, you might or might not be able to literally retrieve it from a file with literally that name. In this case, you can. But when you follow the namespace, that should be enough to get you to the place where you can ask for the file. So in this case, if we go, so because there is an SBOL converted version of the entire iGEM registry on SynBioHub, this namespace of synbiohub.org gives me enough information that I can connect to that with one of the libraries, and I can ask synbiohub.org, give me a copy of BBA J23070. And it will give me a copy of BBA J23070 if I ask for that URI, which has that object and all of its child objects. So BBA J23070, that's our sort of file name that we're looking for. That's why that's the display ID. Synbiohub.org that's my starting point for where I'm going to go to look and ask for this file. And in between is everything else in the URL that wasn't part of the namespace or the display ID.
Jake, would you mind if I add one more thing on that?
Sure.
So the key thing is these are URIs. And so you actually have to, the namespace has to be a place that you own. You can't mint URIs in namespaces like igem.org because those belong to somebody else. They don't belong to the people ourselves actually who created this igem list of parts. So it's just not permissible to use namespaces that are outside of your control, just like you can't create websites in other people's namespaces, even if you felt that might be appropriate.
And importantly, if you go to igem.org and you ask the igem.org web server for this SBOL part, you will be disappointed. Is that clear?
Yeah, thanks for expanding on that.
Welcome. All right, any more questions about the horrible RDF plumbing before we actually get to the data model? A lot of the time, once you've sort of got yourself set up somewhere sane about where you're storing your data, this part of it should mostly disappear from you in the same way that you don't need to know about the JSON that underlies an SVG graphic. This is down in that grimy level. So let's talk about the actual data model itself. I'm going to start here with components, which are in many cases the starting point of any design that we want to talk about. DNA, RNA, protein, small molecules, cells, media recipes, the design for an experimental sample, all of these are components. And a component has at its core two things. One is a type. We have to have at least one type that says which of those types of things is it. Am I talking about a piece of DNA or am I talking about a strain of cells? Role allows me to add additional enhancing information. So for example, if you're talking about a piece of DNA, role is where you put a sequence ontology term to say that this is a promoter, this is a coding sequence, this is a cut site. You can add lots of those, and there are some suggestions about which portions of the systems biology ontology and the NCI thesaurus are good to get types from, and about where in the sequence ontology is a good place to go get roles from. Now, to describe this, we've got sequences and features that talk about the structure of it. So sequence is where you attach, say, your actual nucleic acid sequence for a piece of DNA that you're designing. Features is where you annotate things like, here's the cut site, and I'm making this plasmid by putting this promoter in front of this GFP in front of this terminator. Constraints add our other piece of structural information. When I say something like I'm putting this promoter in front of this coding sequence, that's a topological constraint. If I say I'm going to put this plasmid inside of this cell, that's also a topological constraint. And you can have other types of constraints as well in terms of things like identity. Interactions are how we talk about things like, this protein represses this promoter, this coding sequence produces this protein. And then at the more abstract level, interfaces are how we talk about which portions of the model of the design you're expected to interact with and which ones you aren't. So for example, an interface is how, if you're looking at, say, one of the cello devices, that's how you say, this is the input part, this is the output part of the device. And then models are essentially pointers outbound to elsewhere. So let's look at a simple example of a component. Here is an iGEM component from the 2016 Interlab. This is part number I13504. And I'm showing the identity here in sort of a short form that you can do, where I've got the hash mark has everything up here is the namespace and the local, and I'm just showing the display ID over here. This is our iGEM 2016 Interlab reporter. I've got a little description of what it is, and it's systems biology ontology DNA type with role of engineered region. Engineered region is our sort of catch-all term for systems that aren't just one clearly defined element. So engineered region is typically what you mark anything where you've started putting parts together to make something more complex. So here we've got a ribosome entry site, a coding sequence, and a terminator that we're going to put a bunch of different promoters in front of. So there's our starting point. Any questions so far? All right, and feel free to tap more into the chat as we go. So features, then, let us talk about the structural aspects of a component. There's several different types of features. Subcomponents are reusable parts, like that coding sequence in there, we're going to use as a subcomponent because I've got a GFP part in the iGEM library that is what that GFP is. I can talk about that being used in a bunch of different systems. So subcomponent says I have, you know that GFP design over there, E0040, I'm going to use E0040 here in this design. And why don't we just directly point to it? Why do we have it as a feature? Because I might have another copy of the GFP over there, and I need to be able to distinguish between the GFP I put in this part of the design and the other copy of that GFP coding sequence that I put in another part of the design, because they're in different places. When we want to just mark a feature like a cut site or a scar that really doesn't have anything more to it, we use a sequence feature for that. Sometimes we want to do something kind of like a subcomponent, but it is a placeholder or it only matters in context, and we use a local subcomponent for that. Sometimes we want to point to something outside, like I don't need to make copies of Arabinose in SBOL because there's already a perfectly good definition of Arabinose out in Kebi or PubChem. So an externally defined lets you just use something in somebody else's database. And the last thing is component references let you point down layers in subcomponents. You'll see how that works in a minute. But let's start with a simple aspect with subcomponents. So here is, again, our expression cassette, which is made up of 3 parts, as I said before, the ribosome binding site, the coding sequence, and the terminator. And so we express that by saying, here's my ribosome binding site. It is a subcomponent that is an instance of B0034. The coding sequence is an instance of E0040. And the subcomponent is iGEM's favorite terminator. So at this point, we've now got a specification that says this engineered region up here has 3 things in a bag, but I don't know anything about where they are yet. I don't know about what their sequences are. We're going to need to start adding that information in soon. So let's talk about sequences and locations. That's one way to say where things are. So sequences are pretty simple. A sequence basically is a set of elements that are just some, FASTA type information, and an encoding statement that says what you're looking at. Are you looking at nucleic acid? Are you looking at amino acid? Are you looking, if for chemistry people, are you looking at something smiles or an inchy key? That's a sequence. Locations say where something is on a sequence. So between sequence and location, you can basically encode everything that you would find in a GenBank. So locations always say a location on what sequence, because if you've got, say, a design with 2 plasmids in it, and each plasmid's got a different sequence, and I need to be able to say which plasmid am I talking about. You could be talking forward or reverse. There's a few different classes. You can be talking about ranges, so like where is the GFP? You can be talking about cut sites, like where is that eco-RI actually going to break things? Or you could be talking about referring to the sequence in general. This order field, we won't talk about it much here, but this is what you can use for representing things like introns, insertions, or when a sequence wraps around and crosses the origin. That allows you to be able to describe something as being located, split up between a bunch of different spots. So let's see an example of locating one of these subcomponents. So on this full component, we've got the actual sequence of this full thing, which is of nucleic acid type. And then we can mark a couple of items on there. So for example, there is an assembly scar from the BioBricks assembly that was used to make this, which has a certain location on this component. And that terminator, which has its own sequence, that's a subcomponent, and it has a location at the last 130-ish base pairs of this sequence. So this is one way that we can now say, here is exactly how these pieces get stitched together. Any questions here? Hopefully silence means clarity. Let's talk about the the functional side. Here, you have interactions. This is how we say things like biochemical reactions, genetic production, inhibition, stimulation, so on, degradation. And you have the roles that each species is playing in that. It's the template for genetic production, and it's the product of that production. It's a reactant for a reaction, or it's the product of that reaction. The transcription factor is the inhibitor that represses, through an inhibition type interaction, the inhibited promoter. So to see how that looks, let's talk about the fact that this system here, this component, this chunk of DNA, we don't just want it because it's a chunk of DNA. We want it because this chunk of DNA makes GFP. So we create another component of type protein for the GFP, and we're going to add an interaction in the system that contains both the GFP and the chunk of DNA. And that's going to say that the coding sequence is the template that makes GFP as its product. This also gives us an example of where we want to use component reference, because I don't actually want to say that this whole piece of DNA makes GFP. The ribosome binding site isn't making GFP. The terminator isn't making GFP. It's the coding sequence that's making GFP. And the component reference lets us look down into the subcomponent at the feature of the coding sequence and say, it's the coding sequence inside of this DNA design that makes the GFP. So that's an example of showing a functional interaction. This gives us There's a little arrow on the diagram showing that the coding sequence makes GFP. Constraints are how we talk about things that we don't necessarily have all the sequence information calculated out for. So we've got an open class of constraints. These just essentially say, you know, this subject has this restriction relationship with this object. Like in identity constraints, we can be talking about things being the same as or different from each other, replacing one another. We can have orientation constraints to say things are the same or different orientations from one another. Topological relations, like you know, that a plasmid is going to be contained in a cell that we're transforming, or sequence order, like things that you know, I want my promoter to precede the expression cassette that it's regulating. That last one will be our example here to show how this works. So I've got this system of a expression cassette that makes GFP, and I want to put two different promoters in front of it. So when I want to put J23101 in front of this in order to express this strongly, I'm going to make a component, which is the device that has both in it. It's got two subcomponents, one for the promoter, one for the expression cassette. And going that far, all I've got is I've got a bag with two pieces of DNA. I want to say this promoter goes right in front of this ribosome binding site. And for that, I use a meets constraint. That's a sequence ordering constraint that says it's in front of it with no gap. Can do the same thing over here to make another one that's supposed to express at a medium level using J23106 instead of J23101. And here again, we're using component references to reach down in to say what it is that we're actually linking, because I'm putting the promoter not in front of the GFP, not in front of a generic system that makes GFP. I'm putting the promoter in front of this piece of DNA, and the component reference lets us reach down inside and talk about the chunk of DNA that's part of this system that makes GFP. So this is a lot of the sort of core ideas for describing a design. I'm just going to pause here for a moment for folks to digest and see if anyone wants to ask any more questions before I start moving onward to more complex things that we can talk about now that we can build our system. All right, so the next thing, I'm building a bunch of these designs. So I showed you, you know, number 1 and 2. In fact, in the Interlab, we made 5 different ones. And a collection just lets you put this stuff in a bag and say, here's my collection. It's got a bunch of stuff in it. They might be components. They might be some of the other top levels we haven't talked about much yet. Not much to say there. Collections are easy. Let's talk about another different use of components to talk about not just a piece of DNA, but say a strain that contains a plasmid that contains this piece of DNA. What have I actually done when I transform a strain? Because in the Interlab, we didn't just put DNA fragments into, into the cells to study them. We had E. coli that we were growing to study. It may seem obvious, but we need to add that to our representation. So here we're going to take this device that we talked about before, a piece of DNA regulated by this particular promoter to make GFP at a high level. We're going to put that into a high copy plasmid backbone with chloramphenicol resistance, the PSB1C3 backbone. To do that, we're going to make a local subcomponent that is saying this is the assembled plasmid. I don't really care about it out of context, so that's why I'm using a local subcomponent for it. So this assembled plasmid contains this DNA, and it contains this backbone, and the two of them meet together. So that's how we can describe, we embedded an insert into a backbone to make a plasmid. And then we're going to take that plasmid. That plasmid is the object being contained by the subject, which is an E. coli DH5 alpha strain. Now I have a description for a transformed cell line that contains a plasmid that's made of a backbone that I've put my genetic insert into. Going a little bit more abstract, the other things we might want to say about this, we might want to define what are the inputs and outputs here. In this particular case, this is pretty trivial. We've got one output and it's GFP. That's the thing we want to measure. Doesn't have any inputs because it's constitutive. And this non-directional is for things that are neither input nor output. They may be both or they may be neither. Another thing that we might want to do, I might want to say, hit this up with a differential equation describing the production of GFP that we've modeled in iBiosim. And model basically just gives me a way of making a pointer to somewhere that we're storing that model and saying what sort of model I should find at the other end of it. Now let's talk about some of the other things we may want to say about our system. If we want to be able to talk about numerical information, we use the measure class. This is an import from the ontology of units of measure. And you can add measurements to any type of object via the has-measure relationship. And a measure basically has three things in it. You've got a number, you've got a unit, and you've got a type that tells you what sort of a measurement this is. So, for example, you may have, you may be saying that you have 200 nanograms of DNA is the mass by volume, is the mass of the species in the sample. So there's the systems biology, ontology has a bunch of good terms for talking about these different types of measurements. These three things together basically give you everything you need to describe how a measurement relates to a system. So I see an interesting question in the chat. Is it possible to assert that something doesn't have a property? For example, if I have a cell in which a gene has been knocked out, I don't really know anything else about the cell other than it can't express a particular protein. Depending on how you want to represent it. I can answer this question at a number of different levels. The most basic is actually an important difference between SBOL and plain RDF or plain graph languages for people who care about that level of the plumbing. Plain RDF, you can't actually ever say you don't have a property. You can only say that you don't know if you have a property. You either definitely have a property or you don't know, because there can theoretically be some more triples out there on a server in some other country that you just haven't merged with yet. It makes the reasoning kind of weak. In SBOL, SBOL 3, you are allowed to assume that when you have a top level object, you have all the information about that top level. And if there's conflicting information out there somewhere, it's a version control issue. You shouldn't automatically try to merge it. So you can say that if I'm holding a copy of this component and it doesn't have something in it, then I can definitively say that nope, there aren't any models associated with this because I've got the component at number of models is 0. We can say how to remove something. So I don't have a specific example of it in here, but you can in fact express deletions through the subcomponent mechanism. So I can say, for example, here's the genome, and I specifically remove this section of the genome. So that's actually a very succinct thing to express in SBOL 3. It was hard to do in SBOL 2. It's very succinct in SBOL 3. If you want to talk about something more qualitative, like like the example of a protein knockout, where so if you know the gene knockout, like you did an edit and you remove that gene, you can express that by saying a deletion. If you just know somebody gave you a knockout mouse and that knockout mouse doesn't make some random protein anymore, there's where I would suggest doing it with a measure, where you say that the measure of protein in this mouse is 0, because you don't have enough information to express that at the at the genetic level. So depending on what you're trying to express, there are a lot of good ways to express it, and which of those you choose depends on what is the knowledge that you have that you're trying to express. You can be very, as long as you're able to think precisely about what is it you actually know and what you don't know, you can express a deletion or a removal. Good question. All right, let me give an example of using measures in the design of an experimental sample. So let's say that we're going to culture this transformed E. coli strain in M9 media with some glucose. Then I can attach measures to each of those things to say how much of it there is. So I could put, say, 10,000 cells. That's a measure by count that's expressing the number of entities. SBO has great terms for these things. I'm going to put 10,000, an estimated 10,000 E. coli as my starting population into 200 microliters of volume of M9 media, along with 2.5 milligrams of mass of glucose. And this is the way that we can describe something like the design of a sample. If I wanted to put some inducer in there for an inducible system, I'd add another simple chemical for the inducer, and so on. If I want to talk about having a mixture of cell types, I add more subcomponents for the more cell types that we're adding in. Now, if I'm doing an experiment, though, a lot of times, I'm not just enumerating specific things. I'm saying something like, I want to vary this. So I have, say, all of the combinations of some different media types versus some different carbon sources or something like that. Or something like the talk that we heard for the keynote, talking about a whole bunch of different possible combinations of promoters to sample from. This is what a combinatorial derivation is for. This is how you describe libraries, you describe combinatorial conditions in an experiment. And here you essentially, what a combinatorial derivation has, it has a component, which is going to be a description of the system with some of the parts being blanks that are going to be filled in. And the variable components point to blanks in the templates and give a bunch of options for how to fill them in. So an example here, studying the effects of a carbon source on various different strains. So we'll keep our M9 media, we'll say 10,000 of a particular strain, but rather than actually filling in the strain, I'm going to use a local subcomponent here to put a placeholder variable for strain, which we're going to fill in later. And the same thing for a carbon source. We're going to put in 2.5 milligrams of something, but it's a placeholder to say this is a variable that we're going to set what those 2.5 milligrams will be. And so we can then take that template and put it in a combinatorial derivation for this project varying carbon. We point to the template, and we're going to point to one variable is for the strain. It's pointing to that strain inside of the template. And we'll put any one of those 5 strains in the collection we built earlier into there. And for the carbon source, we're going to pick any one of 4 different sugars to put in as our carbon source. See, in this particular case, we're going to test all the combinations, and we're going to put precisely 1 in of each. You can also have things that are optional. So, 0 or 1, you can have things where you put in combinations. And you can also, rather than doing everything, you can do sampling. Like, again, in the machine learning example in the keynote, to describe that experiment, we would use a sampling strategy rather than an enumeration strategy and draw however many samples they drew. So in order to link together pieces of information across the cycle, like say, the data that we're going to get from this experiment we just described, for that we use provenance. And we've got essentially two types of relationships we can have. The was derived from link is essentially just a simple link that says it came from over there, no explanation, no follow-up questions, or an activity linked with a was generated by, which is essentially a link with an explanation where you can add in information about different roles, parameters, tools, etc., that were involved in getting from point A to point B. And that's what you use for describing, say, the protocol that you used in an experiment to generate data. Speaking of that, 4 top-level classes that go past real fast. An implementation, this is just your hook for saying this is a real-world aliquot that is supposed to be this plasmid, and maybe I quantify and see whether it actually was that plasmid or not. I might run it through a sequencer to find out for real. Experiment classes are really just sort of hooks for making it easy to collect up associated collections of attachments. And attachments are links to data in a format. An attachment is what you use for pointing to a data frame or a flow cytometry file or a microscopy image or a model descriptor for cello. So here's an example of how we use them. So I talked about this sample where we're going to have this transformed cell that we're going to grow in M9 media supplemented with glucose. And if I want to talk about this being in triplicate, that's where I use implementation. I'm going to build 3 instances of it and then say attach data that came out of each of those instances with an attachment. And in fact, we can use these provenance classes to link throughout the design build test loop. So say the transformation that went from my built plasmid into the DH5 alpha in order to actually generate my transformed strain. The culturing that I do on that transformed strain by putting it into the sample design that I've designed in order to actually produce that sample. This is an intermediate stage of the protocol where I actually put this stuff in. And then we run it through some flow cytometry to generate an attachment that's the FCS file with the measurements of the fluorescence of the individual cells. And that completes our tour of all the classes in the SBOL data model. So we pause here again for a moment. We're going to go and exercise some of these classes in a little bit when we go to the hands-on portion of this. But any last questions before we start moving forward towards that?
Yeah, I have a question.
Sure.
So thank you. This is very cool. I'm pretty new to SBOL, but the last slide was kind of interesting, the implementation step and sort of following provenance. It seems like you're almost starting to describe like a protocol, but not quite. Is that right? Like, how does SBOL think about actually doing something?
That is a really good question, and it's something that a number of us have been working on. So SBOL is not enough to describe a protocol, but an activity is the point where we want to hook on a pointer to a description of a protocol. And there is a working group that has been working on a language for describing protocols that in a few weeks at the Combine standards meeting, that's another online meeting with free attendance, that group is planning to kick off the first public discussions of the protocol activity markup language, which we have been working on for doing exactly that as a complementary standard.
Great, thank you.
All right. I'm noticing some nice discussion on the representation of knockouts in the chat. I'll note, by the way, that yes, Goxel's on top of it. I'm not going to need to stir in there. Any other high-level questions or any questions on anything you've seen so far before I'm going to toss two more thoughts and then turn it over to the hands-on side of the tutorial. All right, so a couple other quick thoughts. There is a well-defined mapping between SBOL 2 and SBOL 3, and there are, at this point, converters that go in both directions. This is important because we want to use SBOL 3 as a nice, simple way of describing things, but a lot of the material out there in places like SynBioHub is in SBOL 2 still. So being able to transform it back and forth, really key. I'm not going to explain the details of this. It's in the spec if you really care. At this point, SBOL 3 is up and working in a number of tools. You're going to see some of these today. In general, the recommended starting point for more information about SBOL is at sbolstandard.org. And in a minute, I'm going to take down mine, turn it over to, I believe Brian is going to be the first person talking about the implementation, who will walk you through the tutorial material from here. So Brian, do you want me to stop sharing and you take over, or what's the plan?
I think the plan is Tom will do a brief introduction to the SBOL library, and then he'll turn it over to me to run the hands-on. And in case anybody didn't see, anyone who's interested in doing the hands-on, instructions for how to set up the tutorial environment have been posted in the Slack.
Can someone enable me for screen sharing, please?
I am in the process of doing
cool.
that now.
Thank
I'm
you.
about to make you the host.
All right. Good, thank you. Oh, and now I've lost. Here we go. So can folks see my slides?
You're good, Tom.
Okay, great, thank you. So my name's Tom Mitchell. I'm the principal developer of the SBOL 3 library with help from Brian and Jake. And I'm going to try and take this pretty quickly through so that we can get right to the hands-on. So the evolution of SBOL 3, we started with a C++ library some years ago, and then that was Python bindings were added to the front of that C++ library, and we found that it was challenging to install because you had to have the C++ compiler in order to install it in a Python environment. So we built SBOL 2 to be a pure Python implementation of SBOL 2. And then with the new SBOL 3 standard, we started fresh and have implemented now SBOL 3, and that's what we're going to be introducing today during the beginning of the workshop. And I also want to give a shout out to, there's also this Java version, libSBOL J3, and the author of that implementation, Goxel, is on and in the tutorial. So if there's questions about how to use that, I'm sure he'd be happy to talk to you about that. This just talks about SBOL 3, which you just heard in great detail. A technical note, SBOL 3 actually implements the 3.0.1 specification. There were some things cleaned up from SBOL 3.0, and the 3.0.1 is not quite finalized yet, but it's in the final stages of that, and we expect that later this fall or later this year. And you're welcome to follow along with the SBOL specification. It's all an open process on GitHub. So digging in, for those familiar with Python and programming in Python, we think that PySBOL 3 is going to provide a very familiar Python-like API. Each SBOL 3 entity is represented as a Python class, and all the instance properties on those entities are either native Python types or list-like types for the unbounded or multi-valued properties. And the one change we did make was we adopted the Python snake case. So you'll see here with the underscores, instead of adopting the literal property names from the SBOL 3 specification, because these snake cases are a more familiar way of looking at these symbols from a Python standpoint. So that's one minor difference, but it's a pretty straightforward transformation between the two. And here's just a quick little example, but I'm going to go into some more examples here in a moment. So here is a program to read an SBOL document in PySBOL 3. So we import the package. You can install this package through pip. We're at 1.0 beta 6, I believe now. But basically you create a document here on line 6, and then on line 7, you say read the document. And then on line 9, we're going to dig out one of the elements that was part of that document. We knew ahead of time what we were looking for. There are ways that you can loop through the document and find out what's there as well. And here's an example of that within a component. This circuit component has a set of features, and we loop through that set of features, and we're looking for just subcomponents here on line 16. And if it's a subcomponent, we print out some information about it. And this is just to give you a flavor of how you might navigate some of the objects in an SBOL 3 document. So don't worry too much about the details, just trying to give some flavor. And we also talked about, Jake talked about constraints. Here's looping over all the constraints in this circuit component. And then Jake had this example. So this is an example pulled directly from Jake's slides. And this is the one version of the Python. There's two versions of this that I'm going to show you. This is one version of the Python for how to construct this object in the blue box that Jake had in his slides. Here on line 30, we construct a component object. And then on the next 3 lines, we set the properties on that object to the values we want. And so to see here are the arrows that link it all up. You know, for instance, we want to set the name to this. Well, if you follow down, that's done on line 31. And setting the description, well, that's done on line 32. So this gives you the parallels between Jake's example and the actual code. And this was the way we originally did it. This is still a valid way to do it. But if you know all the values ahead of time when you're about to construct the object, then you can pass all these into the component constructor and give it its name, its description, and its roles through keyword arguments in Python. So sometimes we know all about the object when we're going to build the Python class for it or the Python instance for it. And sometimes we don't know all the values and we're going to build them over time. You can do either with PySBOL 3, whatever fits your particular need at a given time. Oh, here's the arrows again to show you how it links up. These slides are available, by the way, on GitHub. I think Brian sent out the link earlier. So you can look at these slides later. I apologize for going a little bit quickly here. This was a second example from Jake. He had his ribosome binding site, the coding sequence, and the terminator. And here's my version of the code for all 3 of these objects, how we might build these objects. And each of these, I'm going to look at the top left corner here. On line 47 is where we link up this binding site into the features list by appending it to the features list of the parent component. So that's how we've constructed the object on the first 3 lines here. This is how we link it into the data model by adding it to that component. And here's sort of a summary. We've got beta releases out. You can just do pip install SBOL 3. We have an open development process on GitHub. Please, we love feedback. So, feel free to use issues to report bugs or request features or ask questions, whatever you need there. And we welcome contributions as well. And sometimes a bug report is a great contribution for us because you may be using it in a way we didn't anticipate. And so here's the GitHub URL for the repository as well. Any questions before we go on to the hands-on tutorial?
Yeah. Hey, Tom. Could you say a little bit about what kind of validation SBOL3 does?
Yes. 3 does do some validation. We know that the validation is incomplete. Some of the validation rules are hand-coded on some of the classes, and then a whole suite of validation rules are done through a tool called Shackle, which generates triples. It's all done in RDF. That generates rules, and then there's an interpreter to apply those rules on the RDF of SBOL3. So if I were to take a wild stab, I might say that we have about half of the total set of validation rules implemented, somewhere in that neighborhood. I know that Jake has encountered some places where we're not validating things we know we should be. And so he's opened some issues on that based on his work. But there is some validation done there, and there's a call on a document. You can say document.validate, and it'll apply the rules that we do have on the structure that you've got. Does that answer the question?
Yes, and I think in essence by saying the last part, you also answered what I was going to ask, which is when does the validation happen? It sounds like I invoked that on my own using document validate.
I think so. I think that's right. I'd have to be honest with you, I'd have to go look.
That is definitely correct.
PySBOL,
it is definitely correct. It validates precisely when you ask for validate, and it validates entirely locally, unlike PySBOL 2.
Okay, yeah, thanks.
The other thing, just to note what the classes of validation that is or isn't implemented at this point. So in general, essentially any validator that could be built by looking at the ontology is built, because all of that is getting generated by the Shackle rules. So you can think of that as essentially all of the structural, all the syntactic things. You must have a type, that sort of stuff. The things that are not yet implemented tend to be the more subtle semantic things that are like, in a combinatorial derivation, if you point at some things over here, then we want to walk the graph and make sure that you're also pointing at related things over there. So those more non-local rules are the ones that are not yet implemented.
Okay, and so because we're on this topic, earlier in this intro, you described that you basically adopted closed world constraints. Is it the case that the validator takes that? So that's what the validator kind of works with, is a closed world model?
I have,
Yes.
Yes, mostly a closed world model. I have a super specific answer to that, and so I want to look at it to be sure. But when we take the OWL, the OWL file for SBOL3, we apply one of 3 models to that OWL to build the Shackle rules. And it has to do with this open world model, closed world model, and there's one in the middle. And I don't remember which one generated the right set of rules for us. It's either closed or the one in the middle. It is not the one that's the open world model.
Okay, thank you. Oh, and someone pointed us, Jake, thanks, pointed us to the Shackle part. I was wondering how that's spelled. Yeah. Okay.
Yeah, it's short for shape control language, and it's essentially the problem with, so going a little bit nerdy technical here. OWL has a problem. OWL is too weak to be much use because you have to assume that at any point in time you could receive a new triple that you just have to accept. And shackle is a way. It's a stronger reasoning that basically lets you express a large class of things you could ask a Sparkle query about. With
Yeah, thank you.
close field.
All right, good. So Brian, Brian, I can turn it over to you, or I can have these up on the screen.
Let's go ahead and turn it over.
Okay.
Like I said, the instructions are in the Slack channel. And once we get it switched over here, I'll put that slide up for a minute as well. So you may have to make me host now, Tom.
Yeah. Here, I'm gonna, I think I've let you share now. Oh, maybe not.
I think it's working.
Yeah. Beauty.
You can see Jake's slide now.
Yep. Looks good.
All right, so hello everybody. I'm Brian Bartley. I'll be your tour guide on this magical mystery tour. These are some instructions for setting up your environment for the tutorial. It's more or less the same as what I put in the Slack. One difference here is I didn't mention in Slack that if you're used to working in virtual environments and would prefer to do that, you can obviously set up a virtual environment. Also, if you don't want to clone this entire repo, it is a large repo that has a lot of past materials from previous workshops. There's a more self-contained zip file that I posted in the Slack channel. But either way will get you to the same destination. So do I need to give folks a couple of minutes to get things set up? Has everyone that wants to do the hands-on, have you had a chance to install things? If anyone needs some more time, please let me know. And I'll pause for about 30 seconds. You don't necessarily need to do the Jupyter notebook. You could just use your interpreter, use the regular interpreter. Advantage of the notebook is we've got some notes inlined there in with the tutorial. So anyone need a few more minutes? Okay, hearing nothing, I'll go ahead and get started. So the examples that we're going to create today are from Jake's slides. Let me bring them up real fast. So this presentation is available in that zip file. It's also available in the repo if you want to reference it locally. The first example that we're going to build is this part hierarchy. So it was perhaps mentioned before. If not, I will reiterate it. One of the major selling features and advantages of SBOL is that it allows hierarchical parts-based representations that are not necessarily supported by a flat representation like GenBank. So this is a pretty basic and powerful example, pardon me, of what SBOL can do. The other example that we're going to work on is starts with the same construct. That's this component down here in the bottom, but on top of that, we're going to build a system representation that includes the actual function of that circuit. So that will so that system representation will include an interaction that describes, the expected behavior of this circuit. So we've got both the structural layer to the design and the functional layer. So those are the two examples that we'll be working with. I'll go back to my notebook here. So any questions before we dive in? Okay, and Tom or any of the other instructors, if someone raises their hand and I miss it, please feel free to jump in. So the first thing we're going to do, obviously, is import the modules that we'll be working with, and I'm actually going to use the wildcard import, which typically is not considered good practice, but since I'm working interactively here and I don't want to do a lot of extra typing, I'm just going to go ahead and do that wildcard import. The other module that I'm going to import is Tyto, which was mentioned a couple of times. This is take your terms from ontologies. Just an easy way to work with the different ontologies that are integrated with SBOL. So one of the first things we do before we start working on a design, there's some housekeeping things that we have to do. The first thing is to set our namespace. So as Jake was describing in his talk, each URI is created in its own unique namespace, preferably a namespace that you have control over. So for example, I work for a company called BBN, so I'll use the bbn.com domain, and we're a big company, so I'll just say Synbio. This doesn't actually have to correspond to a real URL. Many URIs do resolve to real URLs, but that's not a specific requirement. And then the other housekeeping task is I'm going to create a document. The document is kind of, if I could use an analogy here, it's sort of like a drafting board for an engineer. It's kind of like your canvas that you attach SBOL objects to. So the first thing we're going to do, a real basic task, is we're going to create a component. And the component we're creating is going to be this high-level component that represents the expression cassette. So the first argument is the display ID or the local ID. This will be used to construct the URI of the object after it's created. I'll show you that in a second. The first argument for a component, it has one required argument, which is its types. That's the field that is used to specify what type of molecule you are working with. In this case, we're working with deoxyribonucleic acid. So here I'm using Tyto to access the ontology term. And then the optional arguments, I'll assign those separately. As Tom mentioned, you can use keyword arguments. That's what I'll do in all of the examples afterwards, is I'll tend to use the keyword arguments, but I just wanted to show you that both ways are supported. So the name and description, you know, that's just sort of text-based free text metadata about the component. For roles, that's the field that's used to describe what kind of part you're using or creating. For DNA parts, we use the sequence ontology. And I forget, what is this one? Oh, engineered region. And just to show you what, you know, what these ontology terms look like. I'll print one out. So you can see these Tyto terms, they get converted to URIs. So these are the URIs for these ontology terms. One other thing I guess I want to mention, it does get tiresome writing out deoxyribonucleic acid. So there are some in the SBOL library, there are some built-in constants which do the same thing. Well, I guess I'll just do that. You can see that they're the same. So you can use Tyto or you can use these built-in constants. Then the nice thing about Tyto, if you forget, if you forget a term or you can't remember it exactly, one of the nice things you can do is there are often these browsers on the web where you can go visit the ontology, and you can you can just interactively browse for different terms that are available. Same with the sequence ontology, the Meso browser here. So there's a really rich vocabulary of terms that you can use that are interoperable with SBOL. And so, for our sort of toy examples that we use in this tutorial, we use a rather limited vocabulary of the most common parts, but, you're free to use, that entire vocabulary is at your disposal.
Is Tyto pulling that from the most current release, or is that a cached version of some form?
Yeah, it is. It is pulled from an ontology lookup service. So it, and those are typically kept up to date. So it should be pulling the most recent version. So in this case, Tyto is pulling from OntoB. It also works with the EBI has an ontology lookup service. You can also configure it to use local ontologies if you have a local file. That works as well. Coming back to our design here. So I mentioned the document earlier. That's our canvas. So I've got to remember to add my new component to the document. And this is a step that's really easy to forget. And probably at some point here, I will forget to add one of my components to the document. So if you see me do that, please call me on it. Okay, so that was the first part, the first component. We're also going to create these other components, the RBS, the coding sequence, and the terminator. So I'll just use the built-in PySBOL's built-in constant here, just because it'll save me some typing. And then I can use the keyword arguments as well. What is the keyword argument here, Tom? I think it's RBS, SBO, RBS.
Yeah, I think that's right.
I'll try it and see. Nope. Oh, because it's SO. Sequence ontology, not systems biology ontology. There we go. Okay. Then the GFP gene. So there are just a few of these built-in constants in PySBOL, but these are, the most frequently used parts. All right, I'll pause there for a second. Any questions, or do folks need a little more time to get caught up?
So just, I can probably figure it out on my own, but I'm going to just say it out loud. So I did get an error, an entity with the identity, and then it lists, I used me.com as my domain. B0034 already exists in the document. I imagine that means I added it twice somehow.
Yeah, which if you ran the same cell more than once, that's probably what happened. Which means the second time you ran the cell, if you made any changes to your component, it probably didn't get executed. I actually don't know if that's true.
The first object that was added is still in the document. The second object was not added to the document.
Yeah.
You
So
asked before about identity conflicts, and you just experienced one.
Yeah.
Yes. Yeah. Thanks.
I think if you if you go back up to the top and create document again and walk forward from there, you should be able to walk cleanly through now. I think you just need a new document and then walk through the cells below that.
Got it. Yeah.
This is one of those places where Jupyter gets a little bit weird on you.
The thing that you just described is exactly what happened. Also, I started to use the cheat sheet in order to hurry myself along.
But you bring up a good point, which is a reminder to me that you need to be adding these objects to your document as you go. So you won't, if you forget to add them to the document, you won't catch that right away in most cases. What will happen is at the end, when you try to validate your document, you'll get an error message that will clue you in to the fact that you forgot to add something. Okay, so we've created these components, and now we're going to create the hierarchy out of them. And one thing that I'll remind you of here is that these components are not, the components are not exactly analogous to features in a GenBank file. And the difference is, and I think Jake touched on this, is that these are templates for a component. So, you know, these components might get reused in different contexts. So it's more of a template or a definition for a part rather than a specific feature on a sequence. And when we start talking about features, then we use this subcomponent. So we're going to create these subcomponents, and these subcomponents use these components that we just created as the template or the definition for the subcomponent instance. So here I'm just instantiating a subcomponent, and the template that we're using for it, the template component is B0034. Okay, and then now we'll create the hierarchy. So this gene expression component that we created earlier, we're going to add these subcomponents as features. And it's basically just creating a list. And this looks like, you know, you could, it just looks like creating an attribute value, you know, this looks like ordinary object-oriented programming in Python, but there is behind the scenes here, there is some validation. So if I was putting the wrong type of object in this list, if I wasn't using a list and I just tried to add a singleton object, the PySBOL has some behind the curtains validation that would tell you you're doing something wrong. So that's some feedback that the library will give you to help guide you to make sure you're creating valid data structures.
Brian, could you say, so here you're creating subcomponents out of each of the components that you had. can you explain a little bit why is it that you have to create one subcomponent for each of these? That seems to follow the standard, but feels like something that would have been done behind the scenes, I guess.
So if there were, so in the previous library, PySBOL 2, there were high-level methods. In particular, there was a method called a symbol that did this behind-the-scenes stuff for you. Since PySBOL 3 is newer and just, has been, hasn't been exercised as much, it doesn't have as much of those high-level facilities to kind of make things easier. So you're really really working at the level of the data model. So there's not in a lot of cases, there's not that easy abstraction. So, you're kind of down in the weeds, creating individual objects, each object, and setting the, setting the properties manually. I don't know if that's a satisfactory answer, but and if it answers your question, but
and I can chime in with a little more info as the main implementer. Though we need that bottom data model in a good state before we build that the next layers up, which sort of the macros and being able to manipulate things. So we haven't gotten to that yet because we're not done with that lower layer of stuff. But those kinds of functionality should be able to be layered on top of PySBOL 3, either directly in PySBOL 3 as a library or in other libraries that depend on PySBOL 3. And an example of that is Jake is building an SBOL utilities repository with a bunch of the tools that he has built on top of, among other things, PySBOL 3. That he reuses across a couple of different projects. So there's a bunch of avenues for that. We just haven't explored too far in that space.
Okay, thanks. But
I
just
definitely recommend contributing helper functions as people build them into SBOL utilities.
Okay. But just so that I'm 100% certain, is there any such circumstance where there would not be, or where there would be some other iteration of subcomponents here rather than just a one-to-one?
Yes. So for example, if I am building something that has two different uses of the B0034 RBS for two different functional expression cassettes, then it's not a one-to-one. It may be, two subcomponents that both map to the same component that they're pointing to.
Okay.
Those things actually happen fairly often as soon as you get beyond single promoter systems.
Okay. But I'm sorry to just continue down this path, but so if I'm going to use the same GFB, right, the same E0040, then I will reuse that E0040 multiple times as the same subcomponent within the next layer?
Different subcomponent. Each time you use it, it will be a different subcomponent, but all of those subcomponents will point to E0040 as the thing that they're an instance of. So, think of it as, like if you were looking at a GenBank file and there were two copies of a gene in there, they both might point to the same protein sequence that was describing what that coding sequence mapped to. It's that same sort of relationship.
Okay, yeah, I got it. Thanks.
So it might perhaps have been clearer if I had picked different names for the subcomponents here, just to kind of show you that, you know, each subcomponent is an instance, a particular copy in the design. And it just, in this case, because it's a simple design, we only have one instance of each component. But if it was a more complex construct, we might have multiple instances.
Yeah, perfect. Thanks.
Okay, so now these are just, what we've built is just an abstract, you know, a very abstract parts-based representation of this construct. So now we're going to start filling in some of the sequence details. And to do that, we created a sequence object. Sequence. And I don't think any of the properties, believe it or not, are actually required. But let's go ahead and give it a nucleotide sequence. So the sequence object has this attribute elements. That is where the nucleotide sequence goes. If we were dealing with a protein, it could be a protein sequence. I don't remember if Kebi strings are supported for small molecules. It seems like they were at one time. RNA sequences as well are supported. I could go look up this part on the registry.
Kebi sequences are supported too.
Okay, good to know. I could go look up the actual part sequence on the parts registry, but just in the interest of brevity, I'll just make up a sequence here. Actually, you know what? I should, because we're going to create this range down here. Let me see if I can find the sequence. And then the other field that this sequence object has, is the encoding, which provides the metadata that tells you, or tells your tool how to interpret this string encoding here. So you know, if you were, whether it's a DNA string, a DNA sequence, a protein sequence. And we're using since we're using the IOPAC encoding, we've got a built-in URI that's built into the library. So we're using that URI here. So now I want to associate this sequence. This is an object that lives separately from the abstract part that we created a moment ago. So now I want to associate the two. So there's something you might think is a little curious here. I want to take a second just to point it out and explain it. So a component can support multiple sequences, and you know, one of the reasons that is supported is, you know, consider the case where you've got a high-level design that consists of multiple plasmids, and so, you know, that high-level design, you know, there's not a single sequence that describes that high-level design. You've actually got, you know, if it's a two plasmid system, you've got two sequences that are associated with that component. So that's why we've got some decoupling here between sequence objects and component objects. And again, I've created a new top level object here. So I'm going to add this to my document. Okay, that seemed to go pretty well. So now the next thing is putting base coordinates on features. We'll just do one of the features here. We'll do the terminator. Got to think for a second to remember how to do this.
I'm sorry, I have a question. So you both need to add the sequence as a sequence to I13504 and add the sequence to the document.
Yeah, and Tom, I think that the old library, the PySBOL 2, would, in a lot of cases, it would just automatically add things to the document. So if you forgot, you kind of had that fail-safe. I don't think PySBOL 3 does anything like that yet. Is that correct, Tom?
Yes, that is correct. Everything you're super close to the metal in PySBOL 3 at the moment. So yeah, nothing or very little gets done without your doing it.
Okay, got it.
So if I remember how to do this, I just did this morning, but I may have already forgotten. So if you start to see me going awry here, Jake or Tom, please let me know. So a subcomponent can have a location that describes its location, in the higher level design. And we're using a range object to describe that location. There are other types of locations. I'm trying to remember. There are, I think there are like cut site locations. I think there are cases where you have a single base. I can't remember the classes that represent those cases, but there are other cases besides representing a continuous range. So the first thing, the first argument to this range is that sequence. So it's pointing back to this sequence object we created. And again, the reason we do that is because the top level design could consist of multiple discontiguous sequences. So that's what this this pointer back to the sequence object is doing there. And then we've got the start and the end coordinates. Okay, so far so good. Okay, so we've created this structural representation of the part hierarchy, put some sequence data on it. So now we're going to switch over and we're going to do the functional annotation. We're going to create this interaction now. So this is the part that we created. This is the gene expression construct. Our high-level system representation. It's also got this protein component that we're going to have to create. And then we have this component that represents the high-level system. And so you can see in this system, you've got a protein and you've got the DNA construct. So this is another good example of where you could attach sequences to both of these components, but the system itself has discontiguous sequences. So when we have a system that consists of these multiple discontiguous and interacting parts, the term that is recommended to use to describe that component is this functional entity term from the systems biology ontology. So this system, it's going to have subcomponents for the I13504 expression construct we created earlier. It's also going to have a GFP component that represents the GFP protein. Oops, sorry. So the systems biology term here is polypeptide chain. That's the official term. I think that the built-in constant, we'll look and see if I remember this right, is just SBO protein, same URI. Got to remember to add these to my document. Okay, and this is, we're going to repeat kind of the same recipe that we did earlier when we created a part subpart hierarchy. So we'll create some subcomponents. And then we want to add these subcomponents as features to the high-level system representation. Okay, next we're going to create this component reference. Jake touched on this earlier. This is just basically an object that maps between different levels of a hierarchy. So it's a way of referring to some subcomponent that is further down the hierarchy of components. Actually, call this E0040. And just to make it a little clearer here, I'll use the keyword arguments. So this E0040 is a subcomponent of the expression construct that we created earlier. And the feature that we're reaching down into is the E0040 GFP gene. And this, I believe, I also need to add as a feature. Okay, so we've got kind of these nuts and bolts in place. So now we can put the interaction between the components. create that now. So the reaction that we're the interaction that we're representing is just the gene production. Genetic production is what it's called in systems biology ontology. We're talking about gene expression basically, and we're abstracting away the transcription process. We're just talking about from gene to protein. So the interaction, it's got a type. That type is genetic production. And this interaction has two participants. One is the template, right? That's the E0040 gene. And then it's got the product, the protein that is produced from that template. So we've got so I believe participation is it's kind of a funny term, but I think it's a term that comes from the systems biology ontology. Another way of thinking about it, which might be more clear to some folks, is it's a species. It's a species that participates in an interaction. We're using the SBO term for template here. And the template in this case is the E0040 reference. And then we have the GFP product. GFP subcomponent corresponds to that product. Okay. So that's the representation for the system. So at this point, there's a lot of details to remember. A lot of things a lot of steps that you could leave out and forget. So just some general advice that's natural. It's hard to keep all of this in your head at once. Having diagrams like this is helpful. Referring back to the specification document and those diagrams that Jake covered, being able to read these it is helpful, and it's perfectly natural that the first few times that you do this, that you're kind of going to have to draw things out maybe and work it out on paper before you create it. But it gets easier with time. It's just a skill that you've got to exercise. So the last thing we'll do is write this out to a file. And I think, Tom, correct me if I'm wrong, it should infer the file format based on the file extension. Is that correct?
Yes, I think that's right. It may only be able to do that on read, but let's try it on write. I don't remember if write automatic does that automatic inference or not.
Okay.
Looks like it does.
And just to show you what this looks like, instead of printing it out to a file, I'll just print out the serialization here. I think for this one, I do have to.
Yes, because it can't infer anything.
Because there's no file extension. Yeah. Oh. So this is what an SBOL file looks like. This is using the turtle syntax. We could have printed it out as XML or in triples or the other RDF formats. So don't worry too much if you can't read this, but there should be some things in there that you recognize. So this is representing that object, the system object that we created just a moment ago, with all of its interactions with the GFP production in it. So you can see each of these turtle paragraphs correspond to an object that we've created. And then the last thing I want to show you is the validation. It's a good habit, just as a sanity check, to run the validator. And I think the validation report will give you errors, warnings, and
that's it.
Okay. So okay. And thankfully, everything worked out. Didn't make any critical mistakes, so I don't have any errors. If you are working along and you find some error in there, then you can iterate through those errors in the validation report, and print them out. Of course, this isn't going to do anything for me, but then you'll get you'll basically get the bug report that will tell you, if something is missing or something isn't linked up quite right. So that concludes the hands-on part of this tutorial. Happy to answer any other questions if folks have them. Also, feedback is welcome. What was difficult? How could we make the interface easier to use? That feedback is helpful to us as well. So,
I know there were a lot of steps here. I think of PySBOL 3 as sort of akin to a programming language, and you wouldn't go very far with the programming language before you start to define functions that are related to the work that you're doing in that language, so that you can create things and link things up more readily and repeatedly. And so I think that's part of where we are in terms of the evolution of this particular module, is that we're still pretty early on, and we're trying to get that low level stuff right before we worry about building the higher and higher levels of stuff. So in terms of feedback to us about what all that what's easy or hard is very useful, and suggestions for more of that macro or higher level functionality is useful. Take a look at Jake's SBOL utilities repository as well for more of that high level stuff, that if you were to put some of this work in practice, things that you'd really need to have to make your life easier. You wouldn't stay at the lowest level of a programming language the whole time. Yes, you can loop over all the top level things in the document. There's a document.objects, and that will give you a list of all the things. Yeah, go ahead, Brian.
Oops, sorry. Little trigger happy.
And could you, Brian, would you mind triggering a validation issue just so we can see an example of one?
Can you go behind the scenes and like, go behind the scenes and remove from the sequence, remove the encoding. Can you do that?
Okay.
Just set that to nil, or null, or none, sorry.
Oh.
Ah,
Well, let me do this.
that's probably still okay though. It's got a value. See, the library prevents you from doing certain things as well. I'm trying to think of what a good example here would be for something that's invalid. The only other thing I can think of is reach down into the data model to remove that field by digging around in the properties that underlie the object. I don't know if we want to get that.
Won't let me do that either.
So in some sense, it does do some validation on input as well.
Yeah.
It has an idea of what types of things are allowed in various slots, for instance. Oh, there you go. You did something, Brian.
Trust me, if anybody can mess things up, I can do it.
Nicely done. Yes. Thanks. No, it's very useful to see what an error would look like, or I mean a validation error. And then I guess the kind of, okay, here we go. All right, sequencing code.
So these are these errors created by the shackle validator, Tom?
Certainly the first one definitely is. I believe that the second one is as well. For sure the first one, because that is one of those one of those numerical constraints that sequence is required. Or encoding is required, right?
Yeah. And you can see that error I think is pretty clear, right? Because we set it to an empty string instead of a valid URI. I think that one's pretty clear. The second error is a little more cryptic. The problem is that it's expecting a URI for a component object in this property, in this instance of property. And the one thing it's not telling you here is that the error is occurring in the instance of property, right? That it expects the value of that property to be set to a URI of a component.
Yeah, thank you. Yeah, and I think there's some more questions in the chat.
Yeah. How does it perform validation? Yes, I think it's more like static program analysis. As I talked about earlier in the workshop, there are two kinds of validations. One is more like this static program analysis. We actually serialize behind the scenes when you run validate. For one of the kinds of validation, the shackle-based validation, it serializes the document to triples to the RDF format, and then runs this shackle rule engine. So then it takes those triples and all the shackle rules and executes those shackle rules in a triple store way, and then we get the report out. So that's one side, and that's much more like the static program analysis. The other side are hand-coded rules, where the rules aren't encoded in the ontology, but more involve navigating the graph of objects to say, over here, you need to be referencing something that's already put in this other part of your object hierarchy. Those rules are live code that walks the Python objects to try and find that. And those rules are where we have the most holes in the validation. I haven't written a lot of those rules yet. There's a few just to make sure that it would all fit in, but there's plenty more that need to be done. And that's a good area. If you encounter some of those that don't exist, adding issues will help us to get to them sooner rather than later. Giovanni, I hope that answers your question. If not, please ask. yet. I see your question. It should. The question is, can you append to something that was previously empty? I think it understands that. It should know that that's a list, and you can just append to it. It would have an empty list, a value of an empty list if you don't set it. I'm pretty sure that's the way it works.
Yeah.
I can try and
that.
Okay,
Yeah.
good. Thank you, Brian. How can we grow a skill set that's broad and deep? You know, I think as Brian said, oh, there you go. So C.roles. I'll get to that broad and deep in a second. I'll let Brian finish here. So yet I think that answers your question on the list values. And then broad and deep, you know, to be honest with you, my background is programming. I'm a software engineer. Biology is most definitely, as Brian can attest, biology is most definitely not my thing. I don't know much biology.
Unless you're talking about beer.
But just doing it, you know, writing programs, working with others. I've had a really good time on GitHub, meeting other people who are doing this kind of stuff, taking in their feedback, asking them for critiques, and so on. So I think, and Brian alluded to this too, it's like a muscle, you've got to exercise it. And so using some of these SBOL, the SBOL family of tools, and there's a bunch of them out there on GitHub, using a variety of them and starting to understand the world that SBOL lives in, reading the spec and so on and so forth. I mean, I really did. I have a printed copy of the spec on my desk, and I went through that spec repeatedly as I was implementing the library. So so that helped as well. I hope that, Giovanni, I hope that answers your question there.
I believe this spec includes a lot of examples too, doesn't it?
It absolutely does, and it's actually quite clear in explaining the relationship of the different objects and what are legal values and so on. And if you actually go online and get the latest version of the spec, it has even more information, like all of the validation rules are listed in an appendix. I don't have that copy printed out. I use that one electronically, but yeah, the latest version has a lot of extra information that's really, really helpful too in building your object models.
Yeah, thank you. This was an excellent tutorial. I'll tell you that just in the end there, I was able to get the document printed. So that was that's pretty cool. The quick question I would have though is, so this is a great intro for the programming side. Is there a similar thing where a Excel to SBOL worksheet can be converted to kind of serve as this kind of example?
There is a tool to do that. Jake has the SBOL to Excel, or Excel to it. There's an, I think it's Excel to SBOL, because Jake does a lot of this where he extracts information from Excel documents and builds the SBOL from them and pulls in things from other places. Hang on a second. Let me just search around for a second, and I may be able to find it.
Yet is on the line, isn't she? Isn't Yet?
She may have just dropped off.
Okay, I think Yet works on that spreadsheet tool as well.
Okay, and that's if you guys can send the link later, that's fine too. I'll be on the
I've got it here.
Oh, okay.
There's nice. There's two. Let me just to everyone. You can probably guess which one's which. There's one of them. And there's the other. So both directions are there. I don't know much about the implementation there. But you know, there's probably documentation there and, you know, issues. You know, feel free to use, I don't know how much various folks use GitHub, but Jake asks me a lot of questions by opening an issue and marking it as a question, so that others can see the questions and chime in with the discussion on the questions and so on and so forth. And particularly for any of these libraries, if the documentation is not great and you don't know where to head with it, I think that's a good opportunity to open up an issue and say, hey, the documentation's not great. You got any extra pointers, that kind of thing, or pointing to examples. Jake uses these tools in some automatic pipelines, so you can see examples of how these are actually used in practice. And I believe that the spreadsheets that he uses, the Excel worksheets, are all committed into Git. So you can really follow along with how that's being used.
Thanks very much. Yeah, that's excellent.
Yeah. And I think you may have some of our email addresses. Feel free to give us a holler that way too.
Of course. Thank you.
Any last minute questions? I think we're well over time. So thank you all for sticking around. We really appreciate it. We hope this was valuable and useful to you. We've given a lot of links for how to get in touch if you have any questions or anything. So don't hesitate.
All right. Thank you all.